In this video, we will discuss about one more effect of this mKRAS or mutated KRAS on the TME, that is tumor microenvironment. Here, we will discuss from the aspect of the glycolysis pathway. So this mKRAS or the mutated KRAS in the tumor cells will lead to the increase in the HK12 and lactose dehydrogenase, and this increased HK12 and lactose dehydrogenase will cause increased amount of glucose in the intercellular cells, and that will in the intercellular part of the cells, and that will lead to the increased glycolysis, and the byproduct of this will have this high lactate productions in the tumor microenvironment. This high lactate productions in the tumor microenvironment will cause different conditions, such as first that it will cause is this hypoxia conditions through this HIF-1 alpha signaling pathways, and these hypoxia conditions will favor immune suppressions by promoting this tumor associated macrophages and these regulatory T cells, and these both of these tumor associated macrophages and regulatory T cells will prevent the activated T cells, will prevent or inhibit the activated T cells through different ways. For example, regulatory T cells will release cytokines such as the TGF-beta, whereas tumor associated macrophages will release the cytokines such as VEGF and ARG1. This is some of the examples. Along with this, what will happen? The glycosylations will also increase. How this glycosylation increase? Because of the change in the mKRAS, it will because of the mutated mKRAS, it will lead to the increased or enhanced hexosamine biosynthetic pathways, and this enhanced hexosamine biosynthetic pathways will cause or will lead to the high amount of this UDP GalNAc, which is a precursor for these glycylinations, and because of the increased glycylinations, it will cause the increased glycosylations, and where it will plays a role, it will plays a role that this UDP GalNAc can interact with this fructose 6-phosphate to help enhance this pentose phosphate pathways, and this enhanced pentose phosphate pathways will cause the increased amount of this non-oxidative pentose phosphate pathways. So this is in short term we can understand that it will lead to the enhanced glycosylation. So this is one of the reason why the glycosylations in the tumor or mutated cells is differ from the normal cells, because the glycosylation is a sensitive process. Any changes in the cellular signalings will affect the glycosylations through different ways, and one of the ways is shown here. So and now how these KRAS mutations will inhibit the T cells, that one of the ways is shown on the right hand side, where this KRAS will cause this enhanced glycolysis, and that will lead to the hypoxia conditions, and this hypoxia condition will activate this HIF signaling pathways, and this will lead to the enhanced expression of this PD-L1 and the release of the cytokines VEGF, and this enhanced PD-L1 can interact with this PD-1 on the T cells, and we have understand from our previous videos that when the T cells enters into the tumor microenvironment, then it will also enhance this PD-1 expressions on its surface, and this PD-1 expressions can interact with this PD-L1 ligand on the tumor cell surface, and these interactions will cause reductions in the immune response against the T cells, or you can say reductions in the CD8 plus T cells proliferations, and their cytotoxic effect thereby also been reduced. So this is in brief or some of the main changes that occurs because of the glycolysis changes that arises due to the mutated mKRAS, and the main culprit, or we can say the main reason for this enhanced glycolysis, is the overexpression of this HK12 in the tumor microenvironment and the overexpression of this lactose dehydrogenase. Both of them contribute to the enhanced lactate within the in the tumor. Both of them contribute to the enhanced lactate secretion in the tumor microenvironment, and that contributes towards immune suppression and pro-tumorigenic conditions. So I hope you understood in brief the effect of this mKRAS on the tumor microenvironment. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.